Preterm birth is defined as delivery before 37 weeks of gestation and is the leading cause of neonatal morbidity and mortality worldwide. Preterm birth is preceded by preterm labor, which involves the premature activation of the labor cascade. In late gestation, macrophages are thought to be involved in the pro-inflammatory response that accompanies the process of labor. However, it is unclear whether such a phenomenon is a cause or consequence. In the current study, we hypothesized that macrophages display homeostatic functions during late gestation, similar to those observed in early pregnancy, and that their deficiency will lead to preterm labor and birth. First, we investigated whether macrophages at the maternal-fetal interface display homeostatic phenotypes in late gestation. Flow cytometry was utilized to identify pro-inflammatory and homeostatic macrophage subsets in the decidua from women who underwent spontaneous preterm birth and those who delivered at term. Two macrophage subsets were identified, a pro-inflammatory subset that expresses TNF and a homeostatic subset that displays alternative activation markers such as CD206 and CD209. Women with spontaneous preterm birth had increased pro-inflammatory macrophages, but decreased homeostatic macrophages, showing a shift in macrophage activation status. We also demonstrated that uterine decidual macrophages displayed homeostatic functions in late gestation by increasing the release of anti-inflammatory cytokines, such as interleukin-10, and restricting the release of pro-inflammatory cytokines, such as interleukin-12. Next, we investigated whether the depletion of such homeostatic cells could induce preterm birth. To address this question, we utilized a CD11b DTR model in which the injection of diphtheria toxin causes the depletion of CD11b positive cells, and pregnancy outcomes were evaluated. Depletion of CD11b positive cells induced preterm birth in 75% of cases, and neonates born to CD11b depleted dams died shortly after delivery. Notably, surviving neonates displayed growth restriction. Using flow cytometry, we confirmed maternal macrophage depletion in the peripheral blood, myometrium, and decidua. However, fetal macrophages were unaffected, and placental macrophages were only partially depleted. Maternal macrophage depletion was associated with neutrophilia in the maternal circulation and a neutrophil influx in the myometrium, but not in the decidua nor fetal compartments. In addition, no changes in the total T cell population were observed in the maternal nor fetal compartments. Yet, macrophage depletion induced a pro-inflammatory milieu in the decidual tissues, which was characterized by the upregulation of pro-inflammatory mediators and an increase in leukocyte activation. Next, we investigated whether the adoptive transfer of macrophages could prevent preterm birth and adverse neonatal outcomes. The adoptive transfer of bone marrow-derived macrophages prevented preterm birth but did not fully improve neonatal survival or growth. Therefore, we next investigated whether M2 polarization of macrophages could increase their effectiveness in preventing adverse perinatal outcomes. We utilized M2 polarized macrophages as a treatment to prevent preterm birth induced by intraamniotic inflammation, a model that resembles the clinical scenario of a subset of preterm birth cases. Treatment with M2 polarized macrophages prevented preterm birth by reducing the maternal inflammatory response and increasing the anti-inflammatory cytokine interleukin-10 in the maternal circulation. Importantly, treatment with M2 polarized macrophages prevented neonatal mortality by reducing specific cytokines in the amniotic fluid and reducing fetal inflammation in the brain and lungs. In summary, this study demonstrates a hitherto unappreciated homeostatic role for macrophages in late gestation, exerted by the suppression of pro-inflammatory cytokines to maintain uterine quiescence and sustain pregnancy until term. Consequently, a reduction in homeostatic macrophages can induce the activation of inflammatory pathways that cause preterm birth and fetal injury, leading to growth restriction. This study was published in JCI Insight.